Alright, let's get back to our investigations. Next up is specification number 4, which is all about investigating how wind speed, light intensity, and temperature changes can affect the rate of transpiration. Before we get started, let's go over a few important precautions for this project. First, always keep the plant shoot submerged in water during the experiment. This is to make sure no air gets into the xylem, which could interfere with water uptake. When you're handling the shoot underwater, be gentle, any sudden movement might introduce air bubbles into the xylem. Also, avoid placing the shoot near heat sources like lamps or radiators. Sudden temperature changes can harm the plant or distort our results. Once the potometer is set up, start your timer right away, and make sure each trial lasts the same amount of time, say, 30 minutes, so we can keep things consistent. Do more than one trial for each temperature setting to get reliable results and reduce the effect of any experimental errors. And finally, use clean water in the potometer. Contaminants could affect the plant's transpiration rate. So, we're looking at how different environmental conditions can affect transpiration. The factors we can test include wind speed, light intensity, and temperature. And, to study these effects, we'll be using a special piece of equipment called the potometer. Now, there are actually two main types of potometers, the mass potometer and the bubble potometer. Let's begin with the first instrument we use to study the effect of environmental conditions on transpiration, called the mass potometer. This instrument works by measuring the change in the plant's mass, which reflects the amount of water that has evaporated from its leaves and stem. In the diagram, we can see the main parts: a transpiring plant with a cut shoot, a layer of oil, a conical flask filled with water, an electric balance displaying a reading. This entire arrangement is what we refer to as the mass potometer. Now, let's move on to the second instrument used to investigate how environmental conditions affect transpiration, the bubble potometer. Unlike the mass potometer, this one measures the uptake of water by the plant, which gives us an idea of the volume of water lost through evaporation. If we look at its diagram, the key parts include a transpiring plant with a cut shoot, a reservoir, a rubber bung, an air bubble in the capillary tube, a volume scale, and a beaker of water. This setup allows us to track how far the air bubble moves along the scale, which tells us how much water the plant has taken up.